have a connection. Hello, everyone. I'm here with Chris Venture. He is at the Radian Lab in the MIT Whitehead Institute, and he's going to show us a little bit of what they do with Planaria.
We're with Planaria. They're a freshwater flatworm species. They can regenerate basically any part of the body. So if you cut them into 5 different pieces, they'll regenerate 5 new animals. They have like a brain, an intestine. They're pretty complex in terms of like what organ systems they have, but no matter where you cut them, they regenerate basically exactly what's missing. So if you cut them like in the middle, then the top part will generate a tail, the bottom part will regenerate a head, including a brain, all the neurons, basically the entire nervous system. So we keep them in these incubators. So it's just a normal room temp at 20 degrees Celsius, just to keep it controlled so there's no like huge fluctuations. And so they live in water, so we keep them in these Tupperware containers.
Awesome.
Yeah, just like normal Ziplocs, right?
I'm so excited. There they are, guys.
And so to like move them around, we'll use just these transfer pipettes. And so in 1 second, you can start seeing them move around. Right now, they're kind of like scrunched up.
And you will feed them algae or?
So they're scavengers, so they eat like meat. So we get calf livers, so we'll get like huge, like fresh calf livers. They'll be delivered to the lab. We like chop them up in little pieces and then puree them. So it's like pureed calf liver. So we'll just like drop them into the containers. Any of these have any? Yeah, so you can kind of see like the little food there. Okay. They'll go in and eat it.
So those are bigger.
Yeah, so...
Why are those bigger than the ones that we just saw?
Yeah, so one thing about planarians is they can actively grow and shrink depending on the nutrient availability that they have. And so when you feed them, they grow larger, and if you starve them for weeks on end, they'll start shrinking.
Oh, wow.
So
when they shrink, are they undergoing apoptosis?
Or partly, like they'll undergo apoptosis, they won't make as many, so they have a population of stem cells that are constantly making all the tissue in the animal. Those stem cells will start like shrinking in number as well. And so you basically, when you have less cells or less stem cells, you'll have less tissue being made, and so you'll just get this gradual decrease in size, but they'll stay the same proportion. So they're basically just shrinking proportionally like down, and when you feed them, they'll grow proportionally out. And so some of them are larger, some of them are smaller. So I guess these guys are moving around pretty well now, so you can kind of see they just kind of like glide along the Tupperware. So they secrete some mucus and use that to glide along the bottom.
Okay. Mucilage is what they call it?
Mucus.
Mucus.
Yeah.
And what sort of experiments are you particularly doing for your PhD?
Yeah, so I'm studying. So we just submitted a paper recently, or just had a paper accepted recently, thanks, in Science as well. That we basically did single cell sequencing using DropSeq. It's like a microfluidics-based method where you can basically get all the transcripts that are actively being made in single cells. And so we sequenced 50,000 cells and basically got every cell in the animal. So including cells as rare as like, there's like an anterior pole where it's basically like an organizer for the head. So it basically says this is like where the head is. And it's basically a group of only 10 cells in the head. And out of like an animal that has over 100,000 to 1 million cells. And so we were able to get like basically very rare cells. We also got basically these stem cell population that's like constantly making all the tissues in the animal. And so over time, you're basically getting all the different differentiation states, all the transition states from stem cell to differentiated cell for all the differentiated cells in the animal. So we're able to get this atlas of,
That's a monumental amount of work. Did you dissociate the cells, or how did you pick up one cell at a time?
Yeah, so basically you'll take each of these, you take an animal, we split them up into different sections to try to enrich for like rare cell types in each one of the sections. and you'll chop that up into really tiny pieces, and you can use this like a collagenase mixture, something that'll basically just like break apart the cells.
But it's not trypsin.
It's like similar to trypsin. It's like not, I'd say it's not as harsh, but it's basically just a way to get the cells to disconnect from one another. So you'll get like a single cell suspension, and I don't know how to, you basically pump these cells in through this like device where they get encapsulated in oil droplets. I can show you the setup. You're not going to be able to see exactly everything, but yeah.
Yeah, I know what you mean.
Yeah. So basically you have these pumps.
This is the microfluidic
so these are the
device.
Pumps that do it, I guess. So you have your microscope. You have this tiny device.
A well plate, like a tiny well plate.
Yeah. And so each of these sets of 4 holes. Basically you have tubing, you have syringes that are put into these pumps, and then you'll attach like tiny tubes from the syringes into each of these 4 spots. So you'll pump in cells, you'll pump in an oil, and you'll pump in these little micro beads that basically have spots on them that can capture all the RNA. And so you'll pump everything in together, and so your oil...
You have like a poly A in the bead?
Yeah, it'll be like a poly T, so it'll like contact a poly A. And so then basically you'll encapsulate within the oil like a single cell, a single bead, and then when it's in that droplet, it'll basically lyse open the cell. All the RNA will get trapped onto the bead, and then each of the beads has like a specific sequence on them that will basically barcode it to say that's like I'm different than this one. And so then later on when you go in and like sequence like all of this, you can sequence everything at one time, and then you can computationally say, okay, everything that has this sequence is from this cell, everything that has this sequence is from this cell. So doing that, you can basically sequence thousands of cells all at once, so it makes it really cheap. And so previously it would cost like $5 per cell to sequence, which is like very expensive, but now it's down to like 50 cents or so per cell. And so that's how we're actually able to do this.
And do you get a similar amount of transcripts in each cell type, or do you get some cells that are a lot more active than others?
It probably varies a little bit about in terms of like how many transcripts are being actively expressed in different cell types, though this is like a very small sampling of the transcripts, so you can actually you can tell what a cell is computationally just by like 1,000 transcripts out of like maybe 20, like a little bit of exaggeration I guess, but like a very small subsampling of the total can tell you what a cell is just based off of what those are. And so it's hard to say exactly how many total transcripts are being made per each cell.
No, I know what you mean.
We only took like a small amount from each, I guess.
Yeah.
Yeah.
And so I guess the stem cells, are they really different in profile to, for example, the cells that you were talking about, the organizer cells?
Yeah, so the stem cells express a bunch of genes involved in germ cells. So like in other germ cells, there are a lot of things that are involved in like chromatoid bodies, like PWE, piRNAs.
Oh yeah.
Like I'm gonna explain this like so quickly, but it's like there's a class of things that are all kind of like served as markers for these like stem cells that are not present in the more differentiated cells. And so as the cell begins to differentiate, it'll turn off those genes, and then it'll express certain transcription factors that kind of like lead it to become certain cell fates. And then when those get turned on, it'll turn on more genes that are specific to each type of cell. It's like a neuron versus like an intestine versus something else, like yeah. And so doing that though, you'll get all of those cells, and then you can computationally like cluster them out based off of like what genes they're expressing, so you can tell like what kind of cell they are.
I have 2 more questions.
Yeah.
One of them is, I heard, and I don't know if this is true, that if you want to get more planaria, you can blend it. and then you can. I heard that. Is that true?
No. So you can't just like blend it up and then they won't reform. I think.
Not reform, but like each piece would become a new one, a smaller piece.
Yeah. So if you cut it into like 5 pieces, each of those pieces will become new.
But like
Planaria.
Tinier, much tinier. Meaning, like, I guess that some stems are quite all over the animal, and you could get a new animal from a very small piece of tissue.
You could do a pretty small piece. Yeah, go for it. I don't know how small it would be. Once you get really, really small, they start not doing as well, like with the regeneration. Probably at some small rate they would be able to reform something as long as you have some stem cells. Like one thing you can do, so the cells are pluripotent, so the stem cells, they can make basically any cell type in the body. You can use irradiation to...
So they're not totipotent?
They may be. So I guess this experiment, basically...
That's a big question.
Yeah. So you can take an animal and irradiate it, and so that'll kill all the stem cells in it. And then you can take a single stem cell from another animal, inject it into the irradiated animal, and that will basically expand to repopulate the entire animal that had been irradiated, basically rescuing it from death. And because they're constantly turning over all the tissues, like you can basically take the genotype of this one and turn it into the genotype from this one.
That's amazing. That's Frankensteinian.
Yeah, so like in that you could take a sexual, and so there's a sexual strain that basically reproduces sexually, and another strain that reproduces just asexually. Basically it'll just like rip itself in half to make more of itself. So you can take that asexual stem cell, put it into the sexual animal, and it'll become an asexual animal over time.
That's pretty amazing.
And so to prove totipotency, you would do the opposite, right? You would take the sexual strain and put it into the asexual strain. And then if that was then able to now make sexual organs, then that would prove that they're actually totipotent.
Right, that's totally...
But that experiment wasn't done, so
but you still, you will do it.
It's totipotent, but yeah, for sure it's pluripotent, yeah.
That's amazing. What was the other question I had? Okay, this is the question. It's a very broad question, and I think maybe you get this question from a lot of people. What is it that Planaria has that we don't have?
So I mean, they maintain pluripotent stem cells throughout life. So we have populations of stem cells, but they're pretty localized to like what they can make. So
multipotent
stem cells, yeah. So like your intestines constantly turning over, but those intestinal stem cells only make the cells in the intestine. And so planarians have this like system of keeping pluripotent stem cells throughout their entire life. They also have this like patterning information that's like set up in the body that basically allows the animal to like know what's there, like what's missing, I guess, like when it regenerates. So it's like just signaling molecules that I don't know, maybe you've like your classes learned about. So like WNTs, like basically things that are involved in like development.
Yeah, no, I know. I am going to teach a course next semester, a developmental biology course, and they will be learning about that.
Yeah, they'll learn a lot about
so
that.
That's I love that.
Yeah, so all of those genes, like WNTs, BMPs, FGFs, so like all of these things are set up in these like gradients along the animal. It's like WNTs are really high in the tail and not in the head. And so basically WNT says, okay, this is a tail. Low WNT says this is a head. And when you cut the animal, that gradient reforms to basically match the new size of the animal. So now the top of the tail that like got cut off, that will now be low WNT, and then the bottom will be high WNT. And then as it like regenerates, it'll say what's supposed to go there.
Do you think there's a similarity with the hogs in the patterning, like during development with other, like for example the fruit fly has these like very particular areas where these hogs genes are expressed, and
that it becomes
it becomes like a leg, or
I think it's a similar concept. It's like it's a similar concept. I mean, if you cut off like a limb of something that can't regenerate, it won't like restructure the expression of those genes necessarily. I guess so this is like this active thing that's being like
yes,
maintained,
exactly, totally.
Yeah, so it's like a similar concept.
Right, no, I know what you mean.
Yeah, so I think those two things are like what let it like very well regenerate basically everything. It's the concept of like actively turning over all tissues. Like we don't turn over every single tissue in our body. So yeah.
Well, thank you so much for showing us around.
Yeah, of course.